Hello, everyone, and welcome to my Week 7 presentation on adaptive immunity. In this lesson, we will explore how the human body uses highly specialized immune cells, known as T cells and B cells, to recognize, target, and eliminate harmful infections. My goal is to explain these concepts in a clear and simple way while connecting them to their importance in public health. The human body is constantly exposed to bacteria, viruses, and other microbes. While the innate immune system responds quickly, it is the adaptive immune system that provides a precise and long-lasting defense. This week, we learn how T cells and B cells work together to identify infectious organisms and activate powerful immune responses that help us recover. These two branches form the core of adaptive immunity, which is the focus of today's presentation. By the end of this presentation, you should be able to describe how bacteria causes disease and how our immune system fights back. You will also learn how T cells become activated and how they eliminate infected cells, along with how B cells produce antibodies that neutralize pathogens. Understanding these mechanisms gives us insight why vaccines work and how the body builds long-term immunity. Adaptive immunity is the body's highly specialized defense system that becomes stronger with each exposure to a pathogen. Unlike the innate immune system, which responds the same way to every infection, adaptive immunity tailors its response to the specific invader. It has three hallmark features: specificity, memory, and diversity. Adaptive immunity is divided into two main branches: cell-mediated immunity, which involves T cells, and humoral immunity, which involves B cells and antibodies. T cells must go through a very controlled activation process to avoid mistakes that could harm the body. First, a pathogen is broken down by an antigen-presenting cell, which then displays small pieces of the pathogen to the T cell using structures called MHC molecules. The T cell then receives a second signal called co-stimulation, which confirms the presence of a real threat. Finally, cytokines guide the T cell into becoming exactly the type of cell needed for the infection. This multi-step activation ensures accuracy and prevents autoimmune reactions. There are several types of T cells, each with a specific purpose. Helper T cells, also known as CD4 positive T cells, coordinate the immune response by activating macrophages and B cells. Cytotoxic T cells, or CD8 positive T cells, directly kill infected or cancerous cells. Regulatory T cells help prevent excessive immune responses and protect against autoimmunity. Memory T cells remain in the body long term and enable a much faster response if the same pathogen appears again. Once activated, T cells have several powerful ways to eliminate infections. Cytotoxic T cells can directly destroy infected cells by releasing perforin and granzymes that trigger cell death. Helper T cells improve the killing ability of macrophages while making them more effective at clearing bacteria. T cells also release cytokines that organize and strengthen the overall immune response. These mechanisms are especially important for fighting viruses and infections that live inside our cells. B cells are activated in a slightly different way. They can recognize antigens directly, but they produce the strongest and most effective response when they receive help from T cells. After binding an antigen, the B cell processes it and presents it to a helper T cell. The T cell then provides cytokine signals that allow the B cell to mature into either a plasma cell, which produces large amounts of antibodies, or a memory B cell, which remains in the body for future protection. Antibodies perform several critical tasks in defending the body. They can neutralize viruses and toxins, preventing them from entering our cells. They can coat pathogens to make them easier for phagocytes to ingest and destroy. Antibodies can also activate the complement system, which leads to the breakdown of bacterial cells. Some antibodies work with natural killer cells to destroy infected cells in a process called antibody-dependent cellular cytotoxicity. Finally, IgA antibodies protect mucosal surfaces such as the respiratory and digestive tracts. In summary, humoral immunity involves B cells and the antibodies they produce. This system is especially effective against pathogens that live outside cells, such as bacteria and toxins. Through class switching and affinity maturation, B cells produce highly specialized antibodies that can provide long-lasting protection. This system is also the foundation of how vaccines help the body prepare for future infections. Even though T cells and B cells have different roles, they work together as a coordinated team. Helper T cells are essential for activating B cells, and antibodies help cytotoxic T cells identify infected cells. Both systems generate memory cells that allow the immune system to respond more quickly and effectively if the same pathogen returns. This collaboration results in powerful and long-lasting immunity. The principles of adaptive immunity have major implications for public health. Vaccinations programs rely on the body's ability to produce memory T cells and B cells. Understanding adaptive immunity helps researchers develop new vaccines, improve infection control strategies, and protect vulnerable populations such as young children, older adults, and those with weakened immune systems. These concepts form the foundation of modern disease prevention. To conclude, adaptive immunity is essential for protecting the body from infection. T cells identify and destroy infected cells, while B cells create antibodies that neutralize pathogens. Together, they form a highly specialized defense system that learns and improves over time. Understanding how these systems function not only deepens our appreciation of the immune system, but also strengthens our ability to promote public health and prevent disease.